gene discovery in a fully sequenced genome. Genes are found by scanning the sequence using gene predicting computer programs and assigning putative functions. By searching for similarities in already existing databases. For many organisms under investigation in ecological genomics, no genomic database is available and genes must be identified in other ways. There are some so-called pre-genomic molecular approaches that may be used to identify ecologically important genes in incompletely characterized genomes. From gene product to gene. In some cases, the primary structure of a gene product, a protein, may be the starting point of gene discovery. This holds especially for proteins that can be isolated relatively easily by some marker or bioassay, or proteins that are highly induced by some experimental treatment. As an example, we discussed the isolation of the metal lothionine gene in a species of springtail, Orchesilla cincta, Orchesilla cincta. Hansbergen et al. 1999. Attempts to pick up the gene by polymerase chain reaction using primers from the non-Drosophila mutant sequence remained unsuccessful. Approaches for identifying genes. Two research approaches are commonly used to identify the specific genes involved. These are the candidate gene approach and the genomic screening approach. Candidate gene approach and genomic screening approach. Candidate gene approach. Genes are selected based on their own known or predicted biological function and on their hypothesized relation to the disease or trait. The problem with the candidate gene approach is that it relies on assumptions about the molecular mechanisms underlying the development of a trait. The candidate gene approach can be very time consuming. Chromosomal abnormalities such as deletions, inversions, or translocations in individuals exhibiting a trait as well as animal models mimicking the trait are especially important for a candidate gene approach. They provide clues to the genetic basis of the trait. Genomic screen approach. A genomic screen is a systematic survey in which polymorphic DNA markers evenly spaced along all the chromosomes are used to determine if a marker is inherited along with the trait indicating genetic linkage. Genomic screening does not require prior biological understanding of the pathophysiology of a disease. It requires large sets of data from families containing multiple members who are affected with the trait, and it tends to be the more expensive of the two approaches. Positional cloning. Positional cloning is the process of identifying a diseased gene based on its location on a chromosome. Genetic screening is the initial step of positional cloning. The usual steps are linkage locates a chromosome area. Fine mapping narrows down the initial genomic area to a smaller region. Candidate gene analysis looks for mutations in genes lying in that small area. Before Human Genome Project was completed, such a physical representation was constructed by using a contig. A group of overlapping DNA fragments that together cover the linked region. The contig is a scaffold or platform on which to place genes and other sequences in the correct position. Positional cloning is a laborious process if the region is large and if the genes and polymorphisms making up the contig are not known. The gene identified through positional cloning was the cystic fibrosis transmembrane conductance regulator gene. The gene identified in 1989 regulates chloride ion transport across the plasma membrane and consists of 27 exons spread over a 230 kilobase pair region on chromosome 7.